Science friends, today we are talking about translation, okay? So we have talked about one major type of RNA so far, and that was mRNA. But today we're going to talk about two more types of RNA, rRNA and tRNA. Okay, and so these are really important. They're present in eukaryotes and prokaryotes. So rRNA, I don't really know that there is anything about that in the slides for you to write down, but that stands for ribosomal RNA. Ribosomal RNA. And so here's our ribosome in this picture here. This little brown part is our ribosome. And what I want you to notice about the ribosome is that it's made up of two subunits. Okay, so this is called the large subunit and the small subunit. Sometimes you'll also see them referred to as the 60S and the 40S subunit. But if you know large and small, that is perfectly fine as far as AP is concerned. So they just make up the ribosome, okay? It's a mixture of mRNA and proteins, and it forms the ribosome. The ribosome is made up of RNA and protein mixture, okay? And then the next, which we're going to learn a lot about today, is the transfer RNA, okay? We already know about the mRNA, which is the messenger RNA. We saw that in transcription. It takes the message from the DNA in the nucleus to the ribosome to be able to make the protein, okay? So messenger RNA, ribosomal RNA, and transfer RNA. So the job of transfer RNA, which we're going to start out with, is to transfer. And what we're transferring is these amino acids, okay? So we're doing translation today. We are going from the mRNA to the protein. And this is happening on the ribosome. Remember, this is the big picture of what we're doing today. And so in order to be able to make this protein, we have to read the mRNA and make amino acids first. And you did that in your activity on Thursday when you cut out the little shapes. And each of those shapes represented an amino acid that's going to be strung together to make up a protein. And so the tRNA is what's responsible for bringing the amino acid. So this right here is our tRNA. On the end of the tRNA, you see three bases, and those are referred to as the anticodon, okay? Anticodon is on the tRNA, and it is complementary to the codon on the mRNA. So see here, this codon says GGC, so this one's going to say CCG. This one says UUU, so here we have AAA. This one on the mRNA says UGG, so the anticodon on the tRNA says ACC, okay? So again, it's another triplet. But the tRNA is bringing that amino acid in according to the instructions on the mRNA so that we can grow this polypeptide chain that you see here, okay? So this is kind of a picture or a model of the tRNA. So this is the two-dimensional structure here in picture A. But notice at the bottom you have the anticodon. Also tRNA, it does kind of look like this when you lay it out flat. It has these loop-like structures. Notice that the tRNA, it's also made up of an RNA, so you've got those U's in it. It's a single structure that's folded on itself, and these dots represent hydrogen bonds that help hold those structures together. So if you look at it more three-dimensionally, it looks a little bit more like this. But in our pictures for what we're going to do today, this is the symbol that we have. So we've got our tRNA right here holding on to our anticodon, and then the different amino acids will attach here, okay? So we have 20 different amino acids, so there's 20 total types of tRNA, okay? Now, remember, some amino acids are coded for by different sets of codons. So maybe CCC is serine. I'm not looking at a chart, so I'm just making that up. But maybe also CCG is serine. And so one tRNA is going to carry potentially all the anticodons for serine, okay? So one tRNA that carries serine cannot also carry glycine. It is dedicated to serine. Now, it can carry all of the codes for serine, but it can only work with serine, okay? So that's not super important, but just a little aside there, okay? So let's look at this process. So once you have an amino acid attached onto the end of a tRNA, it's called charged, okay? If it doesn't have an amino acid on the end, it's called uncharged. So to get it from that charged to the uncharged, it takes an enzyme called aminoacyl tRNA synthetase. Again, it ends in ase, so we know it's an enzyme. And so this enzyme matches tRNA up with the correct amino acid, okay? So we've got our amino acid here. We've got our tRNA coming. They both go in there together. It takes a little bit of ATP to make this happen because we're forming a bond here. And so then we end up with our charged tRNA, also referred to as aminoacyl tRNA, okay? That's the same thing. A charged tRNA or a tRNA with an amino acid attached is also referred to as an aminoacyl tRNA, okay? And so that's done by that synthetase enzyme. All right. Now, let's look at this rRNA. There's the notes on it. That ribosomal RNA. So, again, you have that large subunit, that 60S subunit, and the smaller 40S ribosomal subunit. Also on the RNA, you have the rRNA, you have three spots, okay? The E, the P, and the A spot, or the ape spot, if that helps you to remember it. If you're reading it backwards, it's the ape spot. And so we're going to talk about what those do in just a second. You will also see here the mRNA binding site, okay? Now, on the mRNA, remember, before it left the nucleus in transcription, it had some transcriptional modification. Remember, when it got done with transcription, it was pre-mRNA. But then it went through a few steps, at least in eukaryotes it does, to make it, the final mRNA that's going to go into translation. And so if you remember, those steps were adding a 5' cap, adding a poly A tail, and then using the spliceosome to cut out the introns, okay? So this mRNA binding site, it's going to be able to bind to that 5' cap end, okay? We're going to read this from 5' to 3'. So that 5' cap, another benefit of it, in addition to protecting it from degradation in the cytoplasm, is to help with the binding on the ribosome, okay? So it's going to bind there at the mRNA binding site. All right, so let's talk about these three spots. The A spot, which stands for, I wish that I'd written these in order, but whatever, let's just roll with it. The A spot, which stands for aminoacyl tRNA binding site, okay? So remember, an aminoacyl tRNA is a charged charged tRNA, so it's already got the amino acid on it, okay? The P spot is the peptidyl binding site, so that's where you're going to have the growing polypeptide chain, okay? And then the E site is the last one, the exit site. So it's going to read E, P, and A. They're going to come in this way, and they're going to leave that way, okay? They're going to come in this amino acid spot and then leave in the exit spot. All right, so like we had the three stages, initiation, elongation, and termination in transcription, you also have those three stages in translation. So let's talk about initiation. So in initiation, you have to have the mRNA present. That's what we're reading. You have to have the tRNA with the amino acid, so the charged tRNA. And then you have to have both units of the ribosome come together to be able to initiate. And those pieces together are going to be referred to as the translation initiation complex, okay? Here it says the initiator mRNA. Basically, all of that means is the tRNA that carries the anticodon that's complementary to methionine. Remember, methionine is our start codon on the mRNA. Methionine is AUG in the mRNA. So that means our TNA for methionine is going to be CAT, or TAC rather. I wrote that kind of backwards, okay? And so that's our anticodon on the tRNA. And so that's what the initiator tRNA is. It's the one carrying the methionine that's complementary to that start codon. All right. You also have to have some initiation factors, which are just proteins that help hold this complex together. And then one other note is that the amino acids are always added from the amino acid, the amino to the carboxyl end. So remember, we talked about the other day that amino acids have a specific structure. You should be able to draw this. We had a little trouble with it last class. But this is our amino end on the left. Here is our carboxyl end on the right. And so when we add a new amino acid, it's going to be added over here. The amino end is going to attach to this carboxyl. Okay? You're never going to add it on this side. Okay? That would be wrong. You're always going to add from the amino end to the carboxyl end. Okay? All right. So let's look at these three stages of translation again and talk about these sites, these E, P, and A sites a little bit more. So here's our start codon on the mRNA, which is red here. It says AUG. So here's that anticodon, the tRNA right here, and then the anticodon right here that's going to read UAC, okay? And so it comes in here in this A spot first. But actually, since there's nothing in the P spot because it's the first one, it may actually just go ahead and take this P spot. This bond here that's going to happen is a hydrogen bond. And that's important because it's just a temporary bond that's going to be able to break easily, just like it does when we have transcription and replication. Those bonds between those nitrogen bases are always going to be a hydrogen bond. So here we've got our methionine in the P site in our full translation initiation complex, and we're ready to go. Okay, we used a little bit of energy to make this happen. All right. Then in our elongation step is our next step, and that's we're gonna continue to add these amino acids one by one, bringing in these amino acids, reading from the mRNA, and then using GTP for energy. There's a few other proteins that are going to help in this process called elongation factors that are just going to help move this along the ribosome and facilitate this translation process. Okay. So this is super important. We got to understand these E, P, and A steps, okay? So here's our amino acid that's going to come in the A site. That's the aminoacyl site, okay? And so you see here it's sitting right here on the A site. Now, the P site is where you have your growing polypeptide chain. So once something gets in the A site, this guy is going to be transferred on top of here. And you see that here. It's been transferred on top of the A site. Now this P site is empty, okay? So he's going to be kicked over to the E site and kicked out. And this A is going to move over to the P site. Whoops. And that's what we see right here, okay? So now we have our growing polypeptide chain right there in that middle, in that P peptide site, okay? And so then we're going to be ready, if we go back, to accept the next amino acid. The mRNA is going to slide forward, reading from the 5' to the 3' end, and it's going to read this next codon and bring in the corresponding tRNA with the correct amino acid that has the anticodon that matches the codon. Okay? So that is the elongation step. Now, at the end, you have termination. And so termination is signaled by the stop codon. So when this A site, this last piece, gets to that stop codon, there's those 4 stop codons that are on those charts that we've talked about. Then we're going to synthesize some release factors. So that's going to be proteins that are going to cause, hydrolysis between the peptide and the ribosomes and the tRNA, and it's going to cause the mRNA to be released, the polypeptide chain to be freed, and then the whole complex to disassemble, okay? And the mRNA is going to exit, the polypeptide is going to exit via the exit tunnel. That's what this is right here. That can exit via the exit tunnel, and everything will disassemble, okay? Now, okay. Now, let's talk just for a second about polyribosomes. Ribosomes can synthesize, there's lots of different ribosomes. There's thousands of different ribosomes in the cytoplasm. And so ribosomes can synthesize lots of different proteins. They're not specific to a specific protein. And so lots of different proteins can be synthesized at one time. This is constantly happening in the cytoplasm, okay? It's not a one at a time type of thing. So sometimes you'll also have things called a polyribosome. And so that's a series of lots of ribosomes. And so again, it's still reading from the 5' to the 3' end, and the whole strand goes through many different ribosomes. And so as a result, it makes lots of copies of that polypeptide, okay? It's used to make lots of different the same protein, many copies of the same protein quickly. It expedites it, enables the cell to make several copies of a polypeptide quickly, okay? Now, one other thing I want to point out, okay? Remember, in prokaryotes, you may want to make a note of this on your paper. In prokaryotes, there is no nucleus. There's also no transcriptional modification, meaning there's no 5' cap, there's no poly A tail, there's no spliceosomes that cut out the introns. So, as a result, in prokaryotes, transcription and translation can happen simultaneously. I'm going to write that down. Transcription, translation may occur simultaneously. Okay, that's kind of messy. May occur simultaneously. So basically, because we don't have 2 separate locations, you can't do this in a eukaryote because one process happens in the nucleus and the other process happens at the ribosome. But you can do that in a prokaryote because there's no nucleus. It all happens in the cytoplasm. And so after the 5' end finishes with transcription, a ribosome can bind to it on the 5' end and start translating it, while the 3' end is still being fed and being transcribed, okay? So that's an important difference between transcription translation in prokaryotes and in eukaryotes, okay? Now, just like you have transcriptional modification in eukaryotes, you also have translational modification. So some changes that are made in the protein before it can be functional. So the first one is, and we talked about this a little bit in our cells unit. Remember, all proteins are made up of on the ribosome, but there are 2 different places where you can find ribosomes in the cell. Remember, we have free ribosomes that are in the cytoplasm. I think I spelled that wrong. And then we have bound ribosomes that are on the ER, the rough ER. So, what determines where a protein is made? We talked about this in cells. What determined if it's made on a free ribosome or on a bound ribosome? Well, the difference is where the protein is going. So if the protein is supposed to stay in the cytoplasm, then it'll be made on a free ribosome. If the protein is going to be leaving the cell, then it'll be made on a bound ribosome, okay? And so remember, the bound ribosomes are on the ER. They can add, also if it needs any type of modification, if it's going to have a sugar group attached to it or any type of modification that it would get in the rough ER, then it's going to be made on a bound ribosome. But then remember, it goes from the rough ER to the Golgi, and the Golgi is like the post office. It ships it to where it needs to go in the cell, okay? So that's kind of the difference is where they're made. I want to jump here first though. All ribosomes begin as free ribosomes. So they can be free or bound, but all ribosomes actually begin as free ribosomes. And so for example, here's our free ribosome in step 1. Notice that it's out in here in the peachy colored cytoplasm, okay? Well, some of these ribosomes have polypeptides have a signal on them, and that's going to target them to the ER, the rough ER. Okay, so it's a series of amino acids that's going to say, hey, this guy needs to go to the rough ER. He's got some little extra steps that he needs to do to be able to be processed, and so it's got a signal on it that's going to signal that. Okay, now there's other proteins that are available in the cell that recognize those signals. And so when they see that signal, they're going to bind onto that ribosome and drag it over to the rough ER. So let me show you that here. So here's that signal peptide that's released as the ribosome starts reading the mRNA, making this growing polypeptide chain. Here's that signal peptide, this green thing that says, hey, I'm special. I'm made in the ER, okay? So our signal recognition particle is going to bind to that, and it's going to shuttle it over here to the rough ER, okay? Once it attaches it to the rough ER, then the polypeptide is going to be anchored into the rough ER. The signal recognition particle leaves, and the ribosome continues to make this strand, okay? So right here, we push pause. We pause the making of the polypeptide. But then once the SRP left and it gets bound to the rough ER, then the polypeptide continues to be made. And it's going to be made until we reach that stop codon and it gets cut off from the ribosome and the ribosome disassembles. So then that protein, or that polypeptide, when it's finished, it's going to be released or cut off here inside the cytoplasm, inside the ER. Inside the ER. Here we are in the ER. And it's going to go through its folding. Okay? Now, remember, there's those special proteins called chaperonins that help with the folding, that take it from a polypeptide to a functional protein. Okay? And then if it gets any other special additions, then it can be done in the ribosome, in the ER, and then processed via the Golgi and shipped to wherever it needs to go outside of the cell. Okay? So that is the last piece of post-translational modification and targeting the polypeptides to make sure they get in their right spot. Okay? You have a little review question set on Google Classroom and a Google form today just to kind of review some of this material. So if you struggle with any of it, then go back and look over your notes, or of course let me know if you have any questions and we can go over them in class. And you'll have a great day.